properties.
Yeah, thank you so much everyone for the invitation to be here. The meeting has been fantastic and super inspiring, so it's a real pleasure to speak to you today. What I will be covering today are a lot of different important aspects when looking at antibody engineering and design. So on the one hand, I will be focusing on structure prediction. On the other hand, I will try to give an insight on biophysical characterization of antibodies as well as antibody engineering and how all this applies to specificity and consequently antibody-antigen recognition. When I'm talking about antibodies, I'm immediately thinking about antibodies are flexible, antibodies are variable, and actually dynamics is a key part of antibodies function. For example, this is an example of a recent cryo-EM project I'm working on together with Sandhya. She's also a staff scientist in the Ward Lab, where we're looking at IgEs and looking at how flexible are IgEs, how flexible are the arms. And already from cryo-EM, it's apparent that we get different conformational states for the same antibody. And so it's not surprising that also, for example, antibody-antigen recognition can have different conformations. So that, for example, antibody binding can induce a stronger conformational change, for example, also in the antigen. So these are just a few examples of why actually flexibility is a key determinant for antibody function and antigen recognition. And this is where we did then MD simulations to really show like what are these rearrangements, can we capture those? And these are just two structural morphs, but this project was together with Juliana Hahn and Asai, who is actually also a great asset in terms of how to get flexibility analysis from cryomaps. So to go back, when talking about flexibility, we have to be aware that flexibility comes with a timescale. Different rearrangements occur on different timescales, different processes occur on different timescales. And so we're tackling these with MD simulations. Of course, MD simulations have the limitation that that having long timescale MD simulations come with a great computational cost. And so what we're using are different enhanced sampling techniques that allow us to overcome these high energy barriers faster and access higher timescales that reveal functionally relevant conformational changes, such as, for example, CDR loop motions, rearrangements in the antigen binding site, like VHVL dynamics, as well as antibody-antigen binding. And so this is just a schematics of a workflow that we're using. So we're starting off with a crystal structure or a homology model. We perform then an enhanced sampling technique to capture a broader conformational space in a shorter time. However, using enhanced sampling techniques can distort the free energy surface or can result in biases. And so we're clustering the obtained space to then have starting structures for various short MD simulations. And those MD simulations are then unbiased and capture hopefully a bigger space and also a more physical space. And so what our goal is then is to, in the end, have a free energy surface that allows us to reconstruct thermodynamics and kinetics of the rearrangements we observed. Nowadays, we're working on an approach to actually make this workflow more straightforward in a way that we don't have to fine-tune the parameters each time, but that actually a model does this for us. And so SPIP stands for State Predictive Information Bottleneck, meaning that we're trying to infer the slowest movement of the system and thereby predicting the different states from MD. When I'm talking about antibodies, I'm mainly focusing on the antigen binding side. In particular, I'm talking about the CDR loops that are actually responsible for antibody-antigen binding. As mentioned before, our MD simulations are performed in explicit solvation, so we actually get, in the end, hopefully an ensemble in solution, so we can actually also characterize water in the interface, like what are important aspects where solvent plays a role. And so when we started off doing antibody simulations, we were wondering, where should we start? The loops are flexible, but what might be the most critical part of the antibody structure that's the hardest to predict accurately? And so we were focusing on the CDRH3 loop due to its high diversity in sequence and structure, and no canonical cluster structures were available back then to assign accurately. And so we were looking for examples where we had crystal structure information for different CDRH3 loop conformations that have the same sequence. And we found this antibody. This is only one of various cases we looked at, where we have 3 different CDRH3 loop conformations in solution while the antibody sequence is the same. And what you can see is that the antigen binding conformations lie in the dominant minimum in solution, while the apo structure, the unbound structure, lies in a local side minimum. And so the reason for that are actually crystal packing effects. So the tail region of a symmetry-made fab interacts with the CDRH3 loop and thereby distorts its conformation. Another interesting aspect that we observed, these simulations are performed without the antigen. So we were wondering, it's actually really interesting that the binding competent conformation lies in the dominant minimum in solution. And this actually brings me to discuss briefly our views on antigen binding. And so if you bind to a dominant apo conformation in solution, you would call it lock and key. If you bind to an apo conformation within an arbitrary detection limit, and if your structure pre-exists in solution without the presence of the antigen, you would call it conformational selection. And if the binding competent conformation is not there beforehand, you would call it induced fit. However, as a theoretical chemist, I strongly believe that actually all conformations pre-exist, and only if you bind to a rare conformation, it might come with a high penalty in delta G. And so after we were quite confident that we capture a reasonable CDRH3 loop conformation as space, we were actually wondering, can we also characterize the effect of, for example, affinity maturation? What are the structural consequences? And so we investigated various examples. This is also one example shown here, where we looked at the maturation of a germline antibody. And in this case, we focused on the CDRH3 loop, but we actually see this also for whole FVs and FABs, where you see that upon affinity maturation, you have a substantial rigidification of the antigen binding site, where the population shifts towards the binding competent state. So basically, you go from conformational selection towards lock and key binding. And this is just a schematic showing this, where you really see the population shift towards the binding competent matured conformation. And as I mentioned earlier, we see this global rigidification also in a surface plasticity, not only for the FV or for the CDRH3 loop, but this can actually be also observed for whole fabs. And so this brought us to the question, if all loops rigidify and sometimes mutations are only in one single loop, how is this possible? And so we were wondering, all loops might actually be correlated and influence each other. And that's why we decided to describe the antigen binding site as so-called paratope states in solution that actually can shift the relative VHVL orientation towards each other. And so this is one example where you can see different paratope states, different CDR loop conformation as states that actually reveal different relative interdomain orientations. And so it's not really surprising, as I already mentioned, that having the same sequence for one heavy chain, for example, can change when the structure can change, when you have a different light chain germline. And so here you can see it's the identical CDRH3 loop, but it's just a different paired light chain. So also the heavy chain is completely identical, but you find different dominant states in solution, you find different conformations, and also you find differences in crystal structures. And so this brings me also to a study from Charlotte Dean's group, where she actually investigated if the other loops actually influence the CDRH3 loop, and actually she found as well, in line with that the CDR L1, L2, L3 loops actually strongly influence the shape and the conformational diversity of the CDRH3 loop, making it, of course, a bit harder to predict structures because all of them kind of are correlated with each other. And so, in my opinion, antibodies should be considered as conformational ensembles, especially the CDRH3 loop, as it might be hard to find one right conformation for a specific CDRH3 loop sequence. And so this brings me to another aspect we looked at, especially when you look at antibody-antigen docking. What if you have a substantial conformational rearrangement from an apo to a complex structure? If you dock the apo structure, you might have challenges because you will not capture this rearrangement, or if you capture it, you might have a hard time to pick the right conformation. And so we suggest that MD simulations, and especially the respective weights of the conformation, can substantially improve the accuracy of docking, as it facilitates the choices combined with the docking scores. And now this brings me to another critical point. What if we don't have experimental structures? We have to rely on models. And so the breakthroughs in antibody structure prediction in general, in protein structure prediction, have been substantial in the last couple of years. And you almost can follow up with the most recent tools that allow better, faster, more accurate predictions. And so I just want to highlight a few of them. There are a lot more. So we were interested in how well do they perform? How accurate are they? And this is also just a few of them. And what you can see is on the left that actually the overall C-alpha RMSDs of the FV are pretty good. This means that actually all these tools perform really well in predicting the C-alpha positions of the FV. The only difference is the CDRH3 loop. That's, of course, the most challenging part. But overall, all these tools have been trained really well on predicting actual C-alpha positions of antibodies. However, not having the right CDRH3 loop structure has, of course, an effect on, for example, surface properties, such as hydrophobicity, which is a strongly conformation-dependent biophysical property. And so also there, what we have seen is that averaging, for example, ensemble properties, enhances substantially the accuracy of the actual prediction. Another critical aspect is we have found in some of those structures substantial physical inaccuracies. Even though all these tools are really accurate in predicting the C-alpha positions, they have still some challenges in incorporating physics, meaning that the amino acids cis-amide bonds also substantially change your surface, and also MD simulations cannot help you fix those. So when investing in further experiments, please always check your structures carefully for physical inaccuracies. And so we have this tool that allows to do this, but there are, of course, also other tools. This is just a fast Python package where you can actually incorporate it in various workflows and speed up your calculations. So now that we've seen structure prediction works quite well, apart from the CDRH3 loop, and of course antibody CDR loops should be considered as conformational ensembles, wouldn't it be great to use AlphaFold to then get conformational ensembles of antibody CDR loops? And this is something we have been working on for quite a while. Also there, new tools are coming out every other week, different ideas of how to change the underlying multiple sequence alignment in AlphaFold allows to actually capture different conformational states. The question is, are those states actually within the observed structures for antibodies and also for other proteins? Can we capture different conformations? And so the question was also that we asked, what timescales do we capture? Is there kinetic information in there? Do we capture thermodynamics and kinetics of those rearrangements? And so here we looked at a nanobody where we had 2 different crystal structures available, and we also had in-house long timescale MD simulations where we captured the transition from bound to unbound. And so we were wondering, can we use AlphaFold 2 by using different subsampling strategies to predict those ensembles? And in this case, actually, it worked quite nicely. And then we were looking, why did it work so nicely? And actually, a lot of these conformations here for the CDRH1 loop shown were present in the database, and these are projected here into the free energy surface. So compared to the crystal structures that we used as starting structures for MD, we found similar structures in both minima that were deposited in the PDB and available for use. When we are looking at the FV, on the other hand, the FV I showed earlier, where we had 3 different CDRH3 loop conformations available, we see that it does not perform so well. And actually the reason for that is that not a lot of conformation capture in the PDB are available that actually show these transitions and capture these right conformations, meaning that it's very critical that the data is available in the PDB. So actually getting more high quality structures will consequently determine success in predicting different conformational substates. And this brings me now to antibody characterization and engineering. Here I want to highlight a review that was a great collaborative effort with both industry and academia, where we focused on giving an overview of bioinformatics tools, machine learning, and physics-based descriptors on how to assess developability, and also we provide an overview of in vitro developability assessments and cell line engineering methods as an early part of the drug discovery process. Thinking about developability brings me immediately to hydrophobicity. Hydrophobicity is one key determinant for developability, and so we wanted to see everybody uses a different hydrophobicity scale. Does it depend on the problem you're looking for, which hydrophobicity scale you should use? And so, on the one hand, we had 3 data sets available. One publicly available data set from Yain et al. And 2 proprietary data sets from Roche. And we were wondering, how diverse are our data sets even to start with? Do we capture actually the high diverse space that the OAS, for example, has? So we show here in this projection that actually we do see quite diverse antibody sequences, especially in the Yain et al. Dataset. The Roche dataset is less diverse, and so we were quite confident that we have a good representation of the overall antibody space before continuing to with our predictions. And so we, on the one hand, compared different scores. Here I only show the SASA scores. The results are overall similar. You can use the Hayden method, the SASA score. You can only focus on, for example, positive SASA scores. Whatever you feel like you can use. And the question was, how does the choice of hydrophobicity scale and the availability of structural data actually influence the outcome, the prediction of Higgs data in this case, so hydrophobic interaction chromatography. And what you can see immediately is that PDB structures perform better. Homology models perform worse when you have a more diverse data set. But when you have a similar data set, homology models actually don't perform that bad, probably because the errors average out and you don't have like an error addition where you have different inaccuracies summing up. Overall, the Chi-Doolittle scale performs poorly on hydrophobic interaction chromatography data, but for example, there are other metrics like viscosity and so on where some of these hydrophobicity scales might play another role. Another critical aspect, apart from hydrophobicity and also critical surface property, is actually electrostatics. We developed a tool that's called PEP-patch that allows to quantify patches on the protein surface that gives you the most contributing area on your surface to the respective patch and the residues that are responsible for it. And we were wondering whether electrostatics play a role, for example, in predicting antibody pharmacokinetics, such as heparin column retention times. And what you can see here is that actually, independent of the structural prediction tool used, they perform equally good, meaning that electrostatic is a less conformation-dependent property compared to hydrophobicity, which was quite interesting for us, but not so unexpected. But actually, these patches could actually be quite predictive to predict pharmacokinetics of antibodies. And here I'm showing the two outliers, or let's say the 2 points that span the correlation, where you can see the most positive and most negative patches and how they performed in heparin column retention, in terms of heparin column retention times. Another way to map protein properties are so-called co-solvent simulations. In a great collaborative effort with the Stefano Forli lab at Scripps, we developed this tool that allows you to simulate in a fully automated way different co-solvents within your box. So you have your protein and, for example, your antigen, and then you pack each of them in a box and put each solvent that you want in there. You can use Smiles code, so every molecule that you can describe with a Smiles code can be inserted there, and you can look at the interactions with the protein surface over a simulation. And this allows you to get a density estimation on the protein surface that allows you then to map surface properties, such as, which is important for developability. You can characterize effects of charges and solvents on dynamics. Sometimes you have solvent-dependent antibody-antigen binding. You can also analyze that. And of course, you can inform drug design. They have shown that identification of cryptic and allosteric pocket becomes accessible. And also, of course, you can do paratope-epitope mapping. And we found now a way to actually map them on the surface and get the residues that contribute to it. So it's not only a density anymore, but it's now actually we can quantify the surface and the patches, and you can really nicely see here, for example, complementarity between antibody and antigen, and of course this can further inform antibody design or antigen design. And as I mentioned, looking at charges, what are the consequences of having different charges, brings me to another critical aspect in antibody design, which is protonation-dependent antigen recognition. Protonation-dependent antigen recognition can play a big role in, for example, cancer, tumor environments, but also in terms of antibody recycling. And so predicting or getting the idea of which residues contribute most to this for example, pH-dependent binding still remains a big question. And so which tool should I use to protonate my system? Charges, of course, influence substantially your simulation because they're the most long-ranging interactions possible. And so we were wondering how good or what can we actually predict? And this is one example of a collaboration where we looked at a pH-dependent antibody that was histidine-free. Usually everybody targets pH dependence with histidines, but actually you can also do it with, for example, glutamates or aspartates. They are also equally important. The question is just it might be more challenging. And this is one example where no histidines were present, and we wanted to see what happens when we lower the protonation. And we did a constant pH simulation to get a lot of different ideas of which residues might be the most probable to get protonated, which residues are the most important ones in this process. And so we performed a lot of different simulations here. I just showed two as an example where you can see that the experimentally observed decrease in binding affinity upon lowering the pH is actually in line with an increase in entropy between the relative VHVL interdomain orientation. And this made us think actually that probably the VHVL interface is the one to target when designing pH-dependent antibodies. And so in another collaboration with Andreas Lausten's group in Denmark, we looked at an antibody that had identical heavy chains but different light chains. The responsible residue that we identified from crystal structures was identical in both antibodies. So what could be the driving force for pH dependence? One is pH dependent and one is not. What could be the reason for that? And we then performed MD simulations because we couldn't see it from the static crystal structures, and actually found that the excess of water in the interface towards the histidine can enhance or decrease pH dependence. And then we had the chance to also engineer an antibody where we then tried to increase the excess of water in the interface, and found that by mutating bigger residues into smaller residues, and thereby increasing the excess of water, we observed pH-dependent binding, and could make a non-pH dependent antibody pH dependent. And now I'm almost at the end, and I want to show you a very recent example of a workflow we developed where we looked at antibody-antigen recognition, and do we have methods to capture a lot of aspects that are involved in binding. Binding is not only enthalpy. Interactions, of course, play a big role in binding, but there are also a lot of other things like conformational entropy, complex stability, water that play a role in stabilizing a complex and play a role in antigen binding. And so what we were looking at is, by unbinding the antigen from the antibody, what do we see? Do we see differences between a parental and a matured antibody? And in this case, the parental is the 3b4 antibody, and the matured one is the E10 antibody. And what you can see is that when looking at the time evolution, you can see that the parental antibody has way short-lived salt bridges, while the matured one has super long-lived salt bridges. And another super interesting aspect here is that actually the water bridges increase in the matured antibody, meaning that long-ranged water interactions are further stabilized so that the antibody-antigen complex could then consequentially still go back, or that they still interact, which is a key aspect in recognition as well. And then we performed extensive MD simulations on this binding and unbinding pathway, and could see a substantial difference between the parental and the optimized antibody, meaning that the bound state is actually super stabilized in the optimized antibody compared to the parental, where actually an encounter complex is favored. So the unbinding occurs fast, is stabilized via still attached but further away conformation, and then until it's fully unbound. And this is just one of some examples that we looked at, where actually water plays a key role in replacing interactions that are currently destabilized by pulling the antigens apart. And all these trajectories can help us now also to identify and engineer antibodies towards optimizing binding interfaces. And with this, I want to conclude. On the one hand, I hope I could convince you that CDR loops, especially the CDRH3 loop, is highly flexible and dynamic, and actually should be characterized not with a single structure, but actually with an ensemble of structures. The relative VH/VL orientation can interchange actually quite fast, which is also something to take home when you have a predicted structure. Already short MD simulations could help you to optimize the binding interface, especially then also solvent is involved. Affinity maturation, we observed for a couple of cases that we find a local and global rigidification for both antibodies and T-cell receptors. And this brought us then also to the view that antibodies are reveal correlated CDR loops, and so specific framework residues and different heavy light chain pairings are obviously also strongly involving those paratope states in the respective CDR loop conformations. Antibody-antigen docking, of course, can also profit from having the probabilities of the different conformation states to combine with the respective docking scores. Another very important aspect is hydrophobicity is strongly conformation dependent, and by including MD simulations you can actually increase the probability of having the conformations in there that are aggregating, that are contributing to unfavorable hydrophobic properties. And the same holds for PEP-patch, which is a tool where you can also look at hydrophobic surfaces and get patches which can allow you to predict, in our case, antibody pharmacokinetics, but can also be used to predict antibody hydrophobicities in a very fast manner, and it's also freely available. A thing I didn't show today is that we also looked at thermostability of antibodies, where you could see that by pulling apart the relative interdomain orientations of Fabs and CH3 domains, you could actually see a decent trend in thermostability, which is an indicator that thermostability can be a lot, can mean a lot, but this is one of the contributors in melting. And of course, I hope I could convince you that protonation is a critical aspect and strongly influences biophysical properties. And also when you want to engineer and design pH-dependent antibodies, you should not only focus on the CDR loops, but actually focus on the VHVL interface, and not necessarily only on histidines. Top model is a tool that allows you to predict structural inaccuracies in antibody structure models, also as a Python tool, freely available. And I hope I could convince you also that AlphaFold ensemble prediction can be used as a starting point to predict conformational states, but always strongly depends on the available data. If you want to predict a novel protein that nobody has seen before, you will not be lucky, most probably, by predicting different conformational states of this protein. And another important aspect is characterizing antibody binding, antigen binding, and unbinding pathways can further help to understand determinants for molecular recognition and can consequently facilitate antibody design. And with this, I want to thank the fantastic group of Andrew Ward, in particularly Andrew Ward for being such a great mentor, and of course the compute team at Scripps, my former students who did a lot of those groundwork. And I want to thank you all for your attention and all my collaborators for making this happen.
Fantastic, Monica. Thanks for that. We have quite a few questions on the Slido, if you wouldn't mind. The first is, are we over or underestimating the accuracy of CDRH3 by structural prediction programs like IgFold, AlphaFold3, 8-body builder, given the CDRH3 conformational dynamics?
So I think none of those conformations are wrong if they are physically accurate. I think that all of them might be possible, and we haven't done thorough benchmarking, but we're currently doing it by comparing all tools together, mapping them into a free energy surface of a CDRH3 loop and seeing whether all of those actually are possible and are captured. And because this would mean that combining a lot of those different approaches could actually help us to cover already for properties such as hydrophobicity, a reasonable conformational space.
Very diplomatic. We look forward to that. Given unbound structures for an antibody and antigen, how often do you find your MD simulations can give you the correct binding pose?
So the correct binding pose, so we're mainly comparing we're mainly comparing free APO structures, so antibodies free in solution, and there we actually find quite often that the binding competent conformation lies in or close to the dominant minimum in solution, meaning that having additional weights to the different conformations can actually substantially improve antigen binding predictions.
Great. Did you observe CDR3 rigidification as general beneficial principle or just for a selected model?
So I think in the HIV field, there are a lot of studies that show that broadly neutralizing antibody BNAPs follow a different scheme. So actually conformational entropy is beneficial sometimes. So I'm not saying that an increase in affinity is always correlated with a rigidification, but rather specificity. So maturing towards one target usually results in a rigidification.
Fantastic. And finally, are there ML methods to speed up MD simulations?
Yeah, we've tried quite a few, and actually the premise is always that you know what you're looking for. So ideally you know the conformational transition or have it already and use this as an input in your training set, or use this as an input to get then probably faster collective variables and so on. But it usually does not work quite well when you have no transition and only infer from limited sampling data.
Thanks again to Monica.